If you're here, I bet you're the kind of person who likes to get right to the good stuff, right? No time waste with. Well, today we're zeroing in on a pretty big deal, like a real breakthrough. We're talking about Parkinson's disease and how one of the biggest mysteries surrounding it has finally been cracked.
Yeah, and it's a pretty important mystery to solve, because as you probably know, Parkinson's is, well, it's the fastest growing neurodegenerative disease in the entire world. So any progress we can make is a huge deal. It could really make a difference in a lot of people's lives.
Absolutely, no doubt about it. So in this deep dive, we're going to really unpack some fascinating work that's been done by scientists over at WEHI. These guys, they recently published a paper in Science that's revealed some pretty fundamental stuff about a protein called PINK1. And this discovery, well, it could be a real game changer, especially when it comes to how we think about treating Parkinson's.
Okay, so let's cut to the chase here. For over 20 years now, scientists have known that this protein, PINK1, is somehow involved in Parkinson's. But it's been like a puzzle piece we couldn't quite fit in. Imagine trying to understand how an engine works, but you can only see the outside. You know it's important, but you just can't figure out how all the parts inside connect and work together. And that's what's been happening with PINK1. The structure of it, how it interacts with damaged mitochondria. Remember, those are the powerhouses of our cells. And what actually triggers it to do its thing, it's all been a bit of a black box. And this lack of understanding, where it's been a major roadblock in developing really effective therapies, targeted therapies.
So it's like we had this crucial clue, but we didn't have the decoder ring, right? But now are you saying things have changed?
Exactly. What's so amazing here is that these researchers at WHI have done something nobody else has. They've actually managed to determine the structure of human PINK1. And get this, they've also figured out how it binds to those damaged mitochondria we were just talking about.
This is a world first. This is like finding the Rosetta Stone for Parkinson's, right? So for the very first time, we can actually see, like literally visualize what human PINK1 looks like. And we can see how it gets to work on those little energy generators in our cells when they're not working properly. I even saw this incredible image that WHI put out, and it shows two PINK1 proteins attached to a mitochondrion. It's just amazing, after all this time to finally have that visual, it makes it so real.
It absolutely does. It makes it much more tangible. And to really understand why this is such a big deal, why this structural insight is so important, we need to talk about what PINK1 actually does in healthy cells. So picture PINK1 as this, well, imagine it's like a really meticulous quality control inspector. But for your mitochondria, it's constantly going around, checking on these tiny power plants, making sure everything is running smoothly, and identifying any that are, maybe a bit faulty or inefficient. When PINK1 finds a damaged mitochondrion, it basically slaps a molecular tag on it, a protein called ubiquitin. You can think of ubiquitin as like a cellular kick-me sign, specifically for those faulty mitochondria. It's like saying, hey, this one needs to be recycled. And what's so clever is that this tagging system, it's unique to damaged mitochondria. The healthy ones, they don't get touched.
That's a great analogy. So it's like a really precise system for taking out the trash in our cells. But what exactly goes wrong when it comes to Parkinson's?
Right, so this is where things really start to break down. In people who have Parkinson's, especially those who have a mutation in the PINK1 gene itself, this tagging and removal process, it just doesn't work the way it should. It's like the garbage disposal is broken. So instead of being efficiently recycled, the damaged mitochondria start to pile up inside the cell. And these malfunctioning power plants, they become toxic, and eventually they lead to the death of those brain cells. And if you think about it, brain cells, they have these incredibly high energy demands. They're constantly firing and communicating. So any disruption to their power supply, it can have pretty severe consequences. It makes them especially vulnerable in this scenario. And this is actually one of the major factors that contributes to the development of those Parkinson's symptoms that we all know about.
Right. Brain cells need a ton of energy, and if their little power stations are failing and piling up, it's bound to cause some major problems. So how does PINK1 actually carry out this tagging job? This new research has uncovered this four-step process, right?
Yes, exactly. And this is where things get really interesting. This is where we start to see those previously hidden mechanisms. So the researchers, they've outlined these 4 distinct stages in how PINK1 does its thing. The first step is all about sensing the damage. It's like PINK1 has these, well, imagine it has these tiny little sensors that are incredibly sophisticated, and they can detect when a mitochondrion isn't producing enough energy or if some of its internal parts are starting to break down. And the second step, and this was a big mystery before, is that PINK1 actually physically attaches itself to the surface of the damaged mitochondria. Think of it like a key finding its perfect lock. So once it's securely in place, then we move on to step 3. And that's the tagging process we talked about, where PINK1 modifies those ubiquitin molecules. And finally, in the 4th step, those ubiquitin tags that act like a beacon, like a signal flare. And this signal attracts another protein called Parkin. And Parkin's job is to initiate the dismantling and the recycling of that damaged mitochondria. So what's so groundbreaking here is that those first 2 steps, the sensing of the damage and the precise attachment, they were totally a mystery until now.
So now we have this detailed play-by-play, this choreography of PINK1's actions. What does this mean for treating Parkinson's? It feels like we found a really crucial piece of the puzzle.
It's a big deal. Understanding the precise structure of PINK1 and how it gets activated on those damaged mitochondria, it opens up a whole new world of possibilities for drug development. For years, scientists have been talking about targeting PINK1 with medications. It's been a major goal. But without knowing what it looked like, what the target actually was, or how it worked on a molecular level, it was a really tough challenge. It was like shooting an arrow in the dark. But now the research team and other scientists working in this field, they can use this blueprint, this detailed map, to try and identify drugs that can either boost PINK1's activity in people where it's not working well enough, or even correct malfunctions. And this is especially important for people who have those PINK1 mutations, because those mutations are often linked to early onset Parkinson's. So the hope is that if we can target this specific pathway, we might be able to slow down or even stop the progression of the disease in those cases.
Wow, that's incredibly encouraging because, as we've said, Parkinson's is such a difficult condition for so many people. It can be really hard to get a diagnosis, and it affects people in so many different ways. It's not just the tremors that people usually think of, right? I think our sources mentioned something like almost 40 different symptoms, including cognitive problems like difficulty thinking and even speech problems.
Yeah, that's a really important point. You're absolutely right. Parkinson's is an incredibly complex disease. It has so many different facets and such a wide range of symptoms that can really impact a person's day-to-day life, their overall well-being. And the impact is huge. We're talking about just in Australia, over 200,000 people living with Parkinson's. And the economic burden is massive, over $10 billion every year. So any real progress towards more effective treatments, it has the potential to make a huge difference, not just for individuals and their families, but for the whole healthcare system.
It really puts things in perspective, doesn't it? The sheer scale of the problem and how significant this breakthrough really is. And we should also give credit where credit is due to the researchers who made this all happen. The team at WEHI, it includes Professor David Commander, Dr. Sylvie Caligari, Dr. Nicholas Kirk, and Dr. Alyssa Glukova. It sounds like Professor Commander, as head of WEHI's ubiquitin signaling division, he played a really key role, and Dr. Caligari was the lead author on the study.
You got it. Their work together, their collaboration at the WEHI Parkinson's Disease Research Center, it's been absolutely essential to achieving this landmark discovery. And just to remind everyone, these findings, they were published in the journal Science, a very well-respected journal. It was published on March 13th, 2025. And for anyone who really wants to dig into the original research, the DOI is 10.1126science.do6445.
So let's bring it all together, distill it down to the key takeaway here. Researchers have finally cracked the code, solved the structural puzzle of PINK1, and this protein is absolutely essential when we're talking about Parkinson's disease. And they've shown us exactly how it attaches to and gets activated on these damaged mitochondria to start the cell's cleanup process.
Exactly. This understanding, this fundamental knowledge of PINK1's structure and how it works, it's a massive leap forward, a real game changer in our fight against this disease. It gives us a solid foundation to develop those targeted therapies that could, for the first time, really address the root cause of the problem, the actual cellular mechanism of the disease. It offers real hope for more effective treatments in the future, treatments that could actually modify the course of the disease.
Wow. This deep dive into the PINK1 breakthrough has been incredible. Eye-opening, really. It just goes to show how understanding these intricate biological mechanisms, getting down to the cellular level, can open up paths to some truly groundbreaking medical advances. It makes you think about all the other biological mysteries that are still out there, right? What other cellular processes, once we finally understand them, could lead to entirely new approaches to medicine? Approaches that go beyond just managing symptoms and actually change the way we think about treating diseases. It's a pretty amazing thing to think about. A real reason to be hopeful.